everyone, and welcome to this Qlucore Omics Explorer webinar about new version 3.11. My name is Jan Nilsson and I will be your host today. And we'll start by giving a short introduction to the new version, and I will then make demonstrations using a proteomics dataset, a qPCR dataset, and also an Olink dataset. You can read more about the new release at our website. You can see here some links, and you can then watch both videos, download the new release, and also register for other webinars. We have specialized webinars on proteomics, qPCR data, and Olink data also. That is covered during the next month. So welcome to join also the other webinars. The highlights of 3.11 is improved handling of proteomics data, and I will show that during the demonstrations today, but just so that you know, we have added a lot of new functionality related to unnormalized proteomics data. We can handle empty cells and missing values, and also we have added pre-filters. There is a new normalization option, quantile normalization, that you can select if you have unnormalized proteomics data. The data import wizard has been updated also, so that you can import also column or vector-based data files, not just matrix data files. As I mentioned, the qPCR data handling has also been improved. One new option for normalization has been added, housekeeping normalization, which will simplify analysis for those that have unnormalized data. You can also manage replicates in the new version. You can collapse data on the sample side. You have always been able to do that on the variable side, but now also on the sample side. There are some plot improvements that have been added in the new version also. In the PCA plot, there are improved color options and configurations, and speed has also been improved. When we're looking at the two-dimensional plots, the labels and how they are looking has been updated so that you can move them around, change colors, lines, etc. If we look at proteomics data, one of the important new things is the data tab that you can see here below, where we have added quite a lot of new functionality in the 3.11 version. You can see here there are new options for empty cells, pre-filters. We have added a new option to remove variables with certain annotation values. There is a new transform part where you can offset your data. Before you could have thresholding of your data, now you can uplift your data also. There is a new part in identifier and collapse where you can collapse on the sample side, and as I mentioned before, there is now also quantile normalization supported. For qPCR data, we can now handle also unnormalized qPCR data. There is a new normalization option, housekeeping normalization, and I will demonstrate how you can work with that when you have qPCR data. Olink arrays or panels have been very much increased in popularity in the past years. You have been able to import that as matrix data, but now in the new version you can also import data that is vector or column oriented data. And that's the case for some data that has been exported from Olink software. And I will give you an example here with Olink reveal data. And then you can import CSV files in vector or column formats. Some cases you may get something called a pocket file. That file needs to be converted to a CSV file, and then you can import it into Qlucore Omics Explorer. Below here you can see how the data may be oriented here. If you have vector-based data, there is here in this case a column with sample identifiers, and corresponding sample types, which is a sample annotation. Then you have variable information here, Olink IDs, and corresponding UniProt information, which is then a variable annotation. And to the right, you have the NPX data. And in export of Olink data, you will get normalized data, so you don't have to do a normalization afterwards. I will show this also in a demonstration here with a dataset. As I mentioned before, PCA plots have been improved. There are more color options, also improved speed. And if we look at two-dimensional plots, labels, like you can see here below for different samples and so on, you can move them freely around now, change colors and change the dotted lines and so on. So there is a lot of new options there when it comes to labeling. If you have questions, you can contact me afterwards. There are emails here that you can use, and you can of course contact us through the web also. There are different forms that you can use for that purpose. Now I will end the presentation here, and I will instead go to a demonstration of 3 different datasets, and I will start with a proteomics dataset and import it using the wizard. So I will change now from the presentation to the new version here, where you can see here Qlucore Omics Explorer on your screens, hopefully. And you can see it says 3.11 here on top, and I will start by importing a proteomics dataset. It's unnormalized data. I will use the wizard for this. I will go to File, Import or Open with Wizard here, and then I will select a synthetic TXT file with proteomics data. So I will select Open here, and the import wizard starts like this. First question here, what type of data is this? This is other data, it's not raw count data from RNA sequence. So select that, select separator, it's a tab separated file here. Changes there. How is the data organized? Here the data is organized as a matrix where we have got samples as columns and variables as rows. So it's matrix based. Press next. Matrix orientation, treat rows as variables, that's correct. Press next. Data matrix, we start with the top left cell, which is this cell here. I just select it like this, and then press next. One new thing here in 3.11, when you select the last sample and variable, you use the slider to go to the right, but there is now also a slider where you can go to the bottom right. So this is the last sample and last variable. You click there just to mark the data part. Then press next. And then you can import variable annotations. We have got the variable identifier here as protein names. And then we got additional information here as an annotation. Samples, we got them with the sample identifier, and then there is also a treatment here. Press finish. Data is imported, and we can see it here in the middle here. So this is a traditional PCA plot that we can see here. Now, as I mentioned, the data tab has been extended quite a lot, and we'll visit that first. So let's go to the data tab here. First part tells us what type of data. It's other data, it's not count data and genomic data. Empty cells. Here we can now select to replace those empty cells with 0. So let's first go to the method tab here and change to a table view, and then we can see our data. And in some cases you may have data that has been transformed already, but in this case here we will do some changes to the data. So let's go back to the data tab here, and instead of treat as missing, we'll select replace with 0. When we have replaced with 0, that means that we will, for those empty cells, replace the value that we have in the cells with a 0 value. Pre-filters, here you can select if you would like to pre-filter your data here. And in this case here, we have added this part here, remove variables, where, for instance, you can select variable annotation like the protein name, or in this case, another annotation here. And this makes it possible to remove certain variables here in the data set. There are other options. Those are already available in 3.10. This part is new. Won't use it now. Then we have the transform part. Here we could have a threshold our data, meaning that we could have set a minimum value for all the cell values in the data set. Now in this case we'll instead offset our data, so we'll add a data value to all cells in the data set. So let's say that you have some negative values, would like to make them positive and so on, but also then keep the distances. Then you can select to offset your data. So I select offset here, and then you can select the value like 1 or 0.1 or something like that, and then that will be added to all of your cells. You can see this in the table view here. And you can also then select to log transform your data, and this is this checkbox here, and now we have got log transform data in our data set. Identifier and collapse. Here you can select to collapse on the variable side. New is that you can also collapse on your sample side, and you will then automatically create a new data set. So if you have certain replicates and so on, you may want to do this, or there could be other reasons also to collapse on the sample side. We won't do this in this example here. Normalize, that's new. We can now select to normalize the proteomics data also. None here, but you can also select to use either quantile, or if you have qPCR data, you can use housekeeping. I'll select quantile normalization here, and then the data is quantile normalized also. So now I have a data set where I can start work, where I have normalized my data and can work with the statistical tests and so on, just like before. Let's go back to the method tab and look at it in the PCA plot here. And here in statistics and so on, you can select, for instance, do multi-grep ANOVA, select treatment or something like that, and use the sliders and so on to run the tests just in the ordinary fashion. So data tab is very much updated. Now in proteomics data cases, you can do a lot of things that is useful when you have proteomics data. For instance, new ways of handling empty cells, the transform part where you can use offsetting, you can collapse your data, and you can normalize your data using quantile normalization. I will now change to another data set here, so I will close this data set in the log tab here, and use the log tab here, discard, and we are at an empty space again here, and I will now import a qPCR dataset. Go to File here, Open with Wizard here, and then I'll go to the file that I prepared here. A qPCR example, also synthetics data. It's a tab separated text file. I press Open here. The wizard starts. This is other data also. Press Next here. Separator is tab here. Data is organized here also as a matrix. I select matrix here. Press Next. It rows as variables. Yes, we got the variables here as rows. Press Next. Data matrix starts here. Press Next. Go to the right and go to the last cell here. And then select it. Press Next. Select variable annotations, which is in this case just a symbol here. Press Next, and then you can bring in all the information about your samples also, like this. Press Finish. And this is the small data set here, qPCR data and so on. If we go to the variable side, you can see the variables that we have here. And some of those variables can be used for normalization also. So there are some housekeeping genes that we're going to use. If you know them already, you can just add them as a list here, and I prepared it, so I will here import a variable list. You can either do it under File here, or you can use these icons here. I will select this one here, and then select a file, a text file with 3 housekeeping genes. Those are the ones that we're going to use for the normalization. You can also create lists here internally. You can use, for instance, the search here and then create a new list where you put things like the housekeeping genes in that list. Anyhow, I prepared it already. So in the data tab here, we will use that information here, and I will use the normalization part here where I can select now. Now, since the data is not log transform, I can select to do that as the part of the normalization. There are 2 options here, housekeeping and housekeeping geometrical. The last one here can be used if you don't have log transform data. So in this case here, you can select to use that also for normalization. And as before here, there is also the possibility here to do log transformation before if you would like to do so. You can always look at the data in a table view also to get a feeling for your data and see what you have in your data set. See now, let's go here to the data tab again here, method here, housekeeping, and then you can select a list here, and the list that I will select here is the one called housekeeping here for that one. And then you will get some information here. Make sure that you don't have negative values or zeros in the data set. That's not, and that is not already logarithm. And in this case here, it seems like we do have that. Let's go back here in the process here and have a look at the data. Yes, we can see that we do have data that has been are negative here in this case, so probably this data has already been log transformed here. Let's select just housekeeping and not geometric here, and then the file here is housekeeping. So you can always check your data in the data tab before you select if you would like to use housekeeping or housekeeping geometric mean here. And then as before, you can go back to the PCA plot here again, and then you can start running statistical tests here on your data. So you can select, for instance, a 2-group test here using one of the annotations here, like a high dose against a low dose or whatever, and run the test in ordinary fashion. So that was import of qPCR data and also normalization. I will now close this data set also, and the last example I will give to you is an Olink data set. So I will just close here, and then we will import the data using the wizard also. Go to File here, Open with Wizard, and then I will go to the Olink data. And I've selected here a reveal synthetic data file. It's a CSV file here. Press Open here. And in this case here, the data will be organized as columns or vectors. I'll come back to that in the wizard. Press Next here, and we'll have an overview of our data. Type of data, it's Other here, and let's have a look now. We have a number of columns here, so you can see, but there is just one column which is the actual data from the panels, the NPX column here. Now everything is organized then in a column-based fashion. So we got all our variables stacked on top of each other, and you can see the different Olink identifiers here. So they are the same here, but of course then for different samples, as you can see here. So everything is stacked as vectors. Now, other first in the first question, then separator in this case here, and then how is data organized as a vector or column. That's important. Could be matrix data also from Olink panels. Press next. Sample identifier. We got it here to the left. I select that one. So that will be my identifier for the samples. Press next. And then we have sample annotations. In this case, it's just a type here. There could be other things that I would like to import. But in this example, I just select the type. Press next. Variable identifier. Olink identifier here is used for that one. Press next, and then it's variable annotation. I just take the UniProt information. There could be other information that could be useful to import as well. Press next, and then the last thing here is the NPX values that I have here. Press finish. Data is imported, and if we look at the data, we'll go to the Methods tab here and a table view. Then we can see that we have our different samples up here and the variables are here. So we have now reorganized the data into a matrix. It's already normalized data that we have from the Olink panels that we have imported. So let's see now what we have. In many cases, when you have Olink data, you have also some control samples. And in this case here, you can see that we have negative controls, plate controls, and sample controls. So if I go back to the PCA plot, we can see that everything is stacked together here in the middle. So if we remove the control samples, it's probably going to open up the data set. I'll remove the negative control, and you can immediately see that we are now looking at more interesting data. Remove plate control and also sample control. So this is the information now that I'm interested in. So the real samples here. If we go to the variable side here, we have got the identifiers here, and we can also select to have UniProt identifiers. And that's perhaps more interesting here. So in this case here, I will change here in my data tab from Olink identifier to UniProt, like this. And now we can see the identifier is the UniProt names instead. Now, if I would like to remove control identifiers, I can do that also, and they are often available here also within the panels. And I will import here. Let's see if we can do that. I've prepared those that are control variables here in a small file. So take this as import here, and you can see control variables here. Press open here, and it's just 3 of them. You can see them. AMP1, Ext1, Inc1. That's often control variables that you run into when you have your Olink panels. I will remove those also. They are not of interest here. I can do that with a set operation here, where I take the difference here between the data set, which is example reveal here, and the control variables that I have here, and I will create a new list and apply. And we can see that we have now a new list of variables that are just the same as the one up here, but we have removed the control variable. So we'll use that one. So it's only 1044 out of 47 that we're going to use. And we can see here also all of them. Now, if I would like to do some tests here, in this case, I don't have an annotation that could be useful, but I can import one that I've prepared. So I will go here to File and then Import here, and then I can select annotations that I've prepared. I will be using the sample annotations via wizard for this, so you can see it a bit easier. Called groups in this case here. If we look at that, press next here. So what do we have? All our samples, and then I've just grouped them in group 1, group 2, and those that are control. Next, next, identifier, sample ID, next, columns to be used here for import, press finish, and then we have here not only sample type, but the one that I've imported here, groups, where I've got control, group 1 and group 2. Let's color according to that. You can see that control are the ones that I already have removed. So we can see them here. And if I would like to run a statistical test, I can of course do that. So let's say that we would like to run a t-test or something like that in between the two groups, group 1 and group 2. I can set it up here and we can run the test, and we can of course look at the variables here with the p-values, q-values, fold change values, and so on. So it's just to add those columns that we would like to have. We move the slider here, and we can then slide down to the most interesting ones. I can order them by q-value from the lowest one and the increasing downwards here. And then of course I can then create a list of this here, and then I can select to export that. So that was my Olink example here with in this case a vector-based Olink file that I've imported into the program. So now we have been looking at proteomics data from mass spec, we have been looking at qPCR data, and we have been looking at Olink reveal data in this 3.11 introduction. Last thing here that I'm going to show you is that it could be useful perhaps to have a look also at the new possibilities where we can label things and change colors and so on. If I go to the methods tab here, you can see that we have now a sample PCA plot here colored. If I go to the view tab, we can do some changes under plot settings, show here. One thing that we can change now for PCA plots, it's connected to some data point properties, colors, and so on. We can change gradients and so on, perhaps have one color to the next like this, and we can change that one to light yellow, something like that. So you can change colors, and you can also change properties for the data points, how metallic they will be, the different squares, and so forth. So those things can be changed also within the PCA plots to make it sometimes a bit easier to highlight different things. You can of course set up other types of plots here. If we go to the methods tab here and go to all, we can select the volcano plot here. And let's say that we are interested in a few things like this one over here, and we can say that I would like to label here. You can see that the label is there. Now, if I would like to move this around now, I can just do like this, and you can change where the label is and so on. Make it a little bit easier to configure your plots also. And it's possible for those labels here also to change some other stuff related to them. And you can do that in plot settings also for them. So you can select, for instance, the line style and so on. It's solid now. We could change that perhaps to dot or something like that, and then you can see the changes directly in the plot. So that has also been updated in the new release, and that's something that I think can come in handy when you have very many variables or samples and would like to label them and don't want to have overlapping labels within your plots. That's all that I'm going to show you today. You're most welcome to download the 3.11 version. If you're a customer, you can just go to the software part, log in and download. If you are new to our software, you can download a trial license and try it out. Those trial licenses are 10 days, and you can just look into the program, and if you've got questions and so on, you are most welcome to contact me. Thank you very much for attending today. I will now end this presentation. You're most welcome to contact me if you would like to. You can then do it using the contact information here, Jan Nilsson at qlucore.com, jan.nilsson@qlucore.com, or you can contact our support organization as well. Thank you very much for your attention today, and I hope you will join us in our other webinars as well.